Thank you for the kind introduction and the opportunity to share my work today. So for today, I'm going to give you sort of a glimpse of different applications that you can have with long-read sequencing and how essentially you can have much more information from just one sequencing run. So especially, I think the emphasis for today is that we're looking at clinically relevant variants as well. That's why at the end of the day, there's a few interesting tidbits there that we can sort of learn about. Okay, so essentially the overview for the talk is that I'll give an introduction on what is Oxford Nanopore sequencing for anybody who's too new to that. Essentially, the 3 points is to look at variant detection and sort of look at complex variants, especially because I think it's a real strength of Oxford Nanopore sequencing. And lastly, this is the exciting bit for me where we are actually looking at mutational signatures based on long-read sequencing, which is something really new that we are working on. Okay, so for anybody who hasn't seen this diagram, this is actually a really nice way of visualizing Oxford Nanopore sequencing, where you have a protein pore and DNA is thread through the pore. Essentially, what happens is there will be a current change that you can use neural network-based callers to convert between electrical signals to bases. And this is useful because at the end of the day, if you have modified bases, it captures the signal as well, and you'll be able to call that based on the pattern of electrical signals. And what is really useful for this is that essentially what you get is a complete picture of all the variants you can get from one sample. So for the lab, for the work that we do in the lab in Prof. Joanne Yang's lab, essentially we look at the germline mutations of patients with hereditary cancers, and you can get a lot of information just by sequencing a tumor and a normal pair, for example. So you can get germline mutations, you can look at the pattern of mutations or the mutation signatures based on single nucleotide variants, insertions and deletions, you can look at copy number profiles and also get methylation from there as well. So it's really an all-in-one sort of approach to get all the information, all the genetics that you can get from a sample. What is interesting as a computational biologist is that actually you can integrate all that information and then you can get something more out of that. And that's why one of the things that we did in the lab with sort of the current state of long-read sequencing data out there is really to compare Illumina short reads versus what we can get with really nice whole genome sequencing based on ONT technologies. So this is a busy diagram, but essentially it's an IGV screenshot showing you the top panel, which is short-read sequencing based on Illumina in a very problematic region where it's also clinically relevant, where there's a germline predisposition gene in that region as well. In the middle panel, it shows you sort of a phased profile of reads. So you can actually cluster reads based on genotype, and you can identify 2 alleles from the same patient and look at whether any alterations are in one specific allele. What is really exciting to me is that with the same reads, you get methylation information, where you can see sort of a cluster of reads with blue signals where essentially it's hypomethylated. And yeah, it doesn't show up well on the screen, but essentially it corresponds to the CPG site right there as well. So at the end of the day, you can actually use these long reads to span really complex regions, get methylation out of it, and you sort of have a functional readout as well. So at the end of the day, there are many applications to this to look at whether a certain gene is working, whether the patient has a hypomethylated site, for example, and you can use that to have more sort of clinical applications as well. So this is right out of the box. You don't have to do anything with it. But what is really interesting is that there are actually public data sets out there already that we can test our tools and sort of test different methodologies. So this is a really nice study from a group in China, which generated a data set of 22 colorectal cancer samples. It's paired normal sequencing, a sort of older chemistry, R9 chemistry, with a relatively good metrics. So it has read length N50 about 30, and it's actually quite a nice study where the authors actually showed that you can look for variants, especially structural variants, in very important genes in colorectal cancer. But I wanted to take this one step ahead because long-read sequencing isn't just about looking for rearrangements. You can actually push it further and you can actually look at phase information. And this is useful because it tells you that one of the allele, for example, is lost in the patient in the tumor, but it's not lost in the normal sample, normal intestinal tissue. But on the other hand, you can look at germline polymorphisms where you can see the same pattern in both the tumor and normal and additional alterations that happen. So this is another level of detail you can use to identify whether your variants are indeed real or artifactual, for example. And yeah, I think where long-read sequencing really shines is where you can look at complex rearrangements. And in a lot of different cancers, they are actually quite very prone to large-scale rearrangements of megabases, for example. And one particular one that I focus on is extrachromosomal DNA or ecDNA. So one of the interesting biological tidbits about ecDNA is that it doesn't follow sort of the canonical segregation of alleles. So segregation of ecDNA circles is actually, you can think of it as a plasmid. But since it's not tethered by chromosomes, it's randomly segregated. So what you will get is that daughter cells can have different number of circles per cell division, and then you have such a very large variance of the number of copies of ecDNA in a cell. And this is very well captured in the fish image, where essentially you just take two cells and you see the difference in the red signal, which is the signals marking the ecDNA circle. And other biological features include it increases transcription or gene expression of genes, and essentially we think it's a very strong driver of clonal evolution. That's why this is of interest and is something that I'm actively working on. So one of the interesting properties is that we were actually looking at ecDNAs at different stages of the disease, and to our surprise, really, we could actually find it in pre-cancer lesions in Barrett's esophagus, the pre-cancer stage of esophageal cancer. And in this study, we actually look at two different cohorts to show that we actually see ecDNA at a very early stage, and we can actually look at the dynamics of that in even early disease. So in the same data set, what we do with the colorectal cancer samples is to carry out de novo assembly. So a quick primer for anybody really is that if you are doing de novo assembly, you are assembling the reads or the sequences just based on the sequence itself. It's my favorite metaphor really is that you're doing a jigsaw puzzle without a box. So you're just tiling the sequences and trying to match it up, and then assembling it from sequence itself from scratch. And then you get things like squiggles like that, and essentially one of the workflows that I developed is to use the fly assembler, which is a really good short read assembler, but also it works with long reads as well. Yeah. And yeah, to no surprise really, in that 22 CRC cancers, I actually found a case with ecDNA in that region. I assembled it, and it's actually one of the sort of candidates that affected a known oncogene, so the MET oncogene. And in this case, it's actually quite straightforward, where the reads actually span the entire circle with even coverage, and you can actually just identify the circle just based on assembly itself. And I think this is one of the examples where, for example, this is a complex biomarker that different groups are looking at, because it can tell you whether a patient is likely to have more severe disease or resistance to drug therapies specifically. So yeah, to even push it further, so one of the papers that is still under review is to really look at how we can use these assemblies or assembly graphs as references, because there can be really complex events that happen, but it's not even possible to use long reads to resolve any of those. So this is one of the examples where it's a very complex graph. There's a very complex knot there, and we use single-cell sequencing at 2 time points of an organoid to differentiate the different clones that have specific versions of the ecDNA. And I think the really main point really is that the take-home message is that in this organoid especially, you can see a shift in the population of ecDNA circles, because at the later passage, P15 in this case, you actually see a higher amount of ecDNAs at a later time point, and there's a shift in clonal dynamics as well. So by combining long read sequencing and single cell DNA sequencing, you can actually see these dynamics and quantify it using this approach. Yeah. So I think this is sort of the last section where I think it's the most interesting to me, because at the end of the day, I wear a different hat, where I am the co-chair of the ICGC Mutational Signatures Working Group, and we're actually trying to see whether we can look at mutational signatures based on long-read data. So there's actually sort of an assumption that Oxford Nanopore sequencing doesn't have enough sequence quality for you to call variants. But in this exercise, actually, I took Colo 829, which is a really well sequenced sample, a melanoma cell line, for example, and then we tried to compare the variant calls from deep variant in the first panel. Claire 3, which, probably bug is here actually, that's to my surprise. Also with short read callers as well. So in this exercise, what I was trying to do is to figure out whether we can recapitulate the signatures that we find in short reads. And I think the interesting part really is that even with a really crude method of filtering based on deep variant, you get a peak of the melanoma signature, which is 7A in this case. But it's also limited because there's sort of a caller-dependent behavior. So Claire 3 performed really well with 2 different signatures. But out of all the 3 or 4 callers that we tested, there's only one caller, one short read caller that can actually recapitulate all 3 signatures, which was Stroker. So I think this is interesting because at the end of the day, there might be a really specific caller type of behavior. But at the same time, this is still sort of a really it's a start for long read sequencing, because we don't have that many tools for short read variant calling. So I think there's plenty of room if you're interested to write a nanopore-based single nucleotide variant caller. Yeah, sorry about that. And yeah, I think that's the. So essentially, I revisited that same CRC dataset to look at the signatures. So in the paper, they actually identified 5 cases with microsatellite instability. They found out by staining and looking at the mutations in the sample as well. But in this sense, I tried to call, this is an older data set, it's actually older chemistry, and you can actually identify the MSI signatures in the red boxes over here. So SBS6 is one of those, 44 and 21. So essentially, this recapitulates what they found clinically when they actually stained the sample, for example. But the interesting finding really is that if you do the same mutational analysis on all samples, you identified one more sample that had a very interesting phenotype. So this patient actually had a polymerase epsilon mutation that results in hypermutations. So essentially a large number of mutations, and it wasn't captured in the paper at all because they didn't even look at this phenotype, for example. And the interesting part is that clinically there's actually, this set of patients respond better to immune checkpoint inhibitors. So at the end of the day, it's of very much interest that you can use signatures to identify these patients and perhaps suggest different treatments from that. Yeah. Okay, that's why one of the things that I do on my spare time really, or Friday nights, is that essentially we generate synthetic tumors as a community so that we can benchmark different tools as part of the International Cancer Genome Consortium. So one of the things that we try to do is to make realistic tumors that actually look like patient tumors. And this is using different methods of generating synthetic data, and most importantly, we generate noisy tumors that actually look like patient tumors that we sequence. So on the plot on the right, you can actually see this sort of little splotch of like green dots, where essentially it's synthetic tumors if you don't actually have any noise added. So once you add different varying levels of noise, it actually looks closer to the sort of the tumors in purple. And that's why at the end of the day, this helps us give a very good way to see whether tools work in different scenarios, different level of difficulties, and we can set up different scenarios or experiments for testing. So this is a team that is doing the work really, and they're an amazing bunch to work with. So this is work from a really talented undergraduate student, Cheryl, who's in the audience today. Essentially what we want to do is to build a test, a signatures-based test based on long-read sequencing, to look at whether we can look at this really complex phenotype called homologous recombination defect. So usually it happens where a patient has mutations in sort of the BRCA-related genes, or POB2, for example, and a lot of DNA damage is accumulated. And these patients actually respond better to specific therapies like PARP inhibitors. So in this project, really, she modeled the mutation burden across 569 samples in the ICGC breast cancer cohort from the EU, looked at all the signatures and tried to use that to figure out whether we can do targeted sequencing based on mutation burden. And at the end of the day, we sort of have a very nice training set from ICGC and a validation set from a very well-annotated cohort, which is the SCAN-B cohort, which is a Swedish study. And they annotated this triple-negative breast cancers with whether they had HRD, whether they had sort of biallelic loss of any of the genes, hypermethylation, for example. And this is how we generated the synthetic data set. So we have actually 100 true positive sets, so 100 true positive tumors from the positive set, and another 100 samples from the negative set. And we compared 2 different software just to see how well they work, and it's sort of like the first step to see whether this concept works even at all, really. That's why this is the slide that I was working till like quite late last night, because at the end of the day, like Cheryl did really amazing work to show that essentially not all tools are equal. Seq Profiler, it has very much difficulty being like identifying signatures at a really low sort of sequence level, or a very low number of variants, for example. It doesn't do very well compared to another tool called MSeq-Act. So the take-home message for this is that by using patient samples to simulate synthetic data, we can actually have a very objective way of comparing whether tools work. We can use it to develop different techniques and try to see whether we can identify with clinically relevant signatures in patient samples. Is that all? Okay, so that's the conclusion for my talk. Essentially, phasing is important. It helps us with germline variant discovery. We can use assembly methods for complex rearrangements and ecDNA. And to even push it further, you can look at cancer evolution if you have different experimental setups. Lastly, we can actually detect mutational signatures with long reads currently already. And you can also like optimize and benchmark it using synthetic data sets that we generate in ICGC Argo. Thank you for your time.
Thank you, Alvin. Wonderful talk. So we have 5 minutes for Q&A. If you have any questions, please approach to the mic stand. Yes, please go to the mic stand. Is that working? Or you can use mine first.
Okay. I think it's
perfect, cool.
Thank you for the interesting talk. So actually, I want to ask if you can share your experience using the
Oxford Nanopore, okay, yeah.
ONT for the SNP. It's like mutation detection, basically, right? Because our team actually also use a lot on Nanopore, and I mean, we use both Nanopore and short read sequencing, and basically most people would know by now that ONT has quite high error rate. And how, if you can share, like, how do you feel of using that? I mean,
okay,
how reliable is that for?
So I think right now, right now it's, okay, if you're looking at callable regions, which are regions where you usually sort of do sequencing in short read regions, except for maybe homopolymers, the older chemistries don't work so well in homopolymers. But there's actually newer chemistry called the R10 chemistry that works much better already, and there are also different ways that you can call. For example, if you can call like duplex calling with 2 reads, sort of similar to consensus variant calling, that actually works much better. But I think the take home is that if you look at just the callable regions, they're usually quite alright actually. It's also something that I think perhaps it's historical, because like previously there were a lot of reports of really bad sequence qualities, but also with better variant callers, you can actually reliably call variants right now. Yeah.
Okay, but it's actually still have, like, I mean, quite significantly higher and much higher than the short reads, right?
I think, okay, I think, yeah, we have to compare the chemistry. So maybe we can chat offline about like what you're using and specifically like, how to go about it, because I think there's a lot of other tricks to improve the quality of the variant calling as well.
Do you know like how many, like the percentage of how much we can get to polish it until we can get it to like the most reliable we can get?
Yeah, I think that there's a few tricks that you can do. Essentially, you can either change the models, or you can use like super accuracy mode, or if you have newer chemistries, you can do duplex calling. So I think that really improves the sequence quality by quite a lot, I think. It's Q20 if you can get the newest chemistries. Yeah.
Q20, okay. Thank you so much.
Thank you.
Thank you. We still have room for one more question. Yes, please.
Thank you. Sorry to take your time. Thank you for your talk. So I wanted to ask that can we, most mutation tools are being generated for genomics data, as in for cancer. Can we overlay those tools for proteomics data as well? Can we overplot the results?
No, that's a good question. I think based on like current state of things, it's still a no, but it's actually currently under development from Oxford Nanopore that they're trying to do protein sequencing, because using the same approach, if you thread in amino acid sequences, you should be able to get some sort of protein sequence out of that. But I think there's only maybe 2 papers that I see that have already done that already.
And just one more question this. So all the tools as shown in your presentation and generally available online as per my knowledge are being made for cancer studies.
Mhm.
So can we also use these tools for generalized studies for other fungal organisms and other genomic data where mutation signatures could play a role?
Actually, yeah, I think so. I actually have seen some really nice work of mutation signatures in looking at sort of viral evolution. So especially, I think there was a few studies looking at some mutation signatures in COVID, for example, like coronaviruses, which is quite interesting. Yeah.
Thank
Thank
you.
you.